Hi everyone, this is Scott Reeves. This is FSN 328, and let's see. Let me get this set up for recording. Okay, so we're going to go finish up Carb Lecture 1, and I am sitting in one of our big labs, so there might be a little bit of an echo, and I can't promise that no one's going to walk in and interrupt this, but hopefully not. So let's continue on where we left off. We went through the first part of this, and we left off right here, and we talked about tissue-specific metabolism of glucose. So the next thing we're going to do in this process is we're going to look at the structure of the monosaccharides, and we're going to talk about those and why that's important. So if we first look at these are the three main dietary monosaccharides: glucose, galactose, and fructose. And even though they all have 6 carbons, they're all C6H2Os basically, they have different structures. And because they have different structures, they are recognized as different molecules by things like transport proteins and enzymes, and each of these also has different taste properties. And the taste properties, for example, in the taste buds, proteins recognize the difference in structure, and I think we all have gathered that fructose is the sweetest of the monosaccharides, and it's one of the reasons why it ends up being so common in certain foods. It's why they developed high fructose corn syrup, which is essentially, high fructose corn syrup is corn starch, and we know corn starch is all glucose, and the corn starch is converted to a corn syrup, and then the corn syrup, which is all glucose, is treated with enzymes and then also acids to break the starch apart, and then the enzymes can convert glucose to fructose. And probably the most common form of high fructose corn syrup is one called HCFS 55, which means it's 55% fructose and 45% glucose. So it's similar in the ratio of fructose to glucose than sugar, which we know as sucrose, which is 50% fructose, 50% glucose. So you can see it's even a little bit sweeter than regular sugar. So, and it's relatively cheap to make. So that's some of the reasons why. It's actually, I have in my notes here, it's almost twice as sweet as sucrose, the fructose is. So when you have that high fructose corn syrup, it's cheap to make, it's a cheap sweetener, it's relatively stable, so it ends up in a lot of different foods. And that's one of the main issues with high fructose corn syrup, is it's in so many things, it can cause fructose levels to be relatively high. And obviously it's a simple carbohydrate, so it's not ideal. So we're going to look at the structures of these, and because they affect the way they're metabolized also. So if you just look at the structure of glucose and galactose, you can see they're both 6-membered ring structures. And that hydroxyl group on carbon number 4 for glucose, you can see that hydroxyl group is down. And if you look at that same part of the molecule for galactose, the hydroxyl group is up. That's the only difference between glucose and galactose. And yet, in many circumstances, they are metabolized differently just because of that one simple change or alteration in structure. If you look at fructose, it's a 5-membered ring. I think we all remember that, but it still has 6 carbons. So in this case, in fructose, there's an oxygen bridge between carbons 5 and 2. And we'll talk about why that happens in a second. Glucose and galactose, the oxygen bridge is between carbon 6 and 1. So that's why that's different. Sorry, carbon 5 and 1. Sorry, for glucose and galactose. All right, so let's look at these. And there's similar, this diagram's out of your textbook. And some nomenclature right away. We see that it has anomeric carbon. We'll talk about what that means in a minute. And also says aldehyde, and hopefully you realize that's misspelled. It's not a hide like an animal hide. It should be Hyde. There's an aldehyde group, and the aldehyde group is CH, double bond oxygen. That's an aldehyde. And if you look over to the right side of this diagram, you see ketone. And in this case, we can see fructose as a ketone instead of an aldehyde. And the ketone, remember, is the C double bond to oxygen with a chemical group on either side of that carbon number 2 in this case. So fructose has a ketone group, and glucose and galactose have an aldehyde group. And that's unique features between those different monosaccharides. It's the location of that double bond oxygen in the overall structure that makes the ring structures form differently, either a 6-membered or a 5-membered, so we'll talk about that. So more nomenclature here. If we look down to towards the bottom of this, we can see the D and the L. And the D and the L represent different enantiomers, right? Each molecule, in this case, can have a mirror image. We refer to that structure of the molecule either as a D or an L. So if we look at this D-glucose structure here, remember this structure, we call it the Fischer structure. Sometimes it's called the open chain or the linear structure. Basically, in this case, it's a linear molecule. And that allows us to see the orientation of the different chemical groups. In reality, in solution, these molecules are going to be in their cyclic structure. But they can open and close within solution as well, but they'll typically always be in their closed structure. But if we look at this, we can see, if you look at the D and the L, for example, if you look at the D sequence here, you can see, for example, the hydroxyl group on carbon number 2 is to the right. Well, on L-glucose is to the left. On number 3, it's the OH is to the left, and on the L, the OH group is to the right. In other words, each of these chemical groups are flipped to the opposite side. And what that does is that causes them to rotate light in the opposite direction. The way that we determine whether or not it's an L or a D is based on the location of the hydroxyl group on the highest numbered chiral carbon. And you can see those hydroxyl groups are denoted or indicated by being in red. So let's review what a chiral carbon is. Remember, chiral carbon is one that has 4 different or unique chemical groups attached. Remember, carbons have 4 covalent bonds. Oxygen will form 2 covalent bonds typically, and hydrogens will form 1. So when you're drawing out these structures, use those general rules so you can look back at your structure and make sure that those rules have been applied in your structure. That'll help you remember how to draw them. But if we go through here and we say, well, let's look at that for the highest numbered chiral carbon. Well, number 1 is not even a chiral carbon because it doesn't have 4 unique chemical groups. It only has 3. If you look at carbon number 2, it's a chiral carbon because it has unique structure here. There's a hydrogen, there's a hydroxyl group, and the rest of the molecule. So 2 is a chiral carbon, so is 3, so is 4, so is 5. 6 is not because it has 2 hydrogens. So the highest number chiral carbon is 5. The hydroxyl group in this structure is to the right. If the hydroxyl group is to the right, it's the D form. If it's to the left, it's the L form. That's as much as we need to know at this point. The other important item that I want to mention again, you probably heard this in biochemistry already, for carbohydrates, for these monosaccharides, it's the D form that is the most common in nature, and it is the one that we metabolize. In other words, it's the functional form. Our body really doesn't metabolize or recognize the L form. So that's an important point to remember. Now if we look at fructose, the same general rule applies. The number 5 carbon is the highest numbered chiral carbon for fructose. In this case, the hydroxyl group is to the right, and in this case, the hydroxyl group is to the left, so that makes it the L enantiomer form. So I'm going to make this simple. If I ask you to draw structures of these monosaccharides, I will say draw it in the Fisher or open chain structure, and I'll always ask you to draw the D forms. So it's important you understand the difference between the D and the L, but you only have to memorize or be able to draw it in the D form. Okay? So let's go from this linear structure to a cyclic structure. And this is in your textbook. This is, I think, from edition 6, I believe. But there is a typo in this one, of course. That's what makes learning structures even more fun, is when there are mistakes in the textbook. But you can see here the Fisher projection here. Here's the 3 carbon. They put a hydrogen here, but it had to be a hydroxyl group. So that's all that means. I tried to add that in there so you can see. So an easy way to do it is just remember that structure, practice drawing that structure for D-glucose. And then let's look at, here's D-galactose here. So remember that hydroxyl group on carbon number 3 is just flipped. Sorry, carbon number 4 is flipped. And then here's fructose in the linear structure. Now what this is showing is going from the Fisher projection to a cyclized form into a form that we can call the Haworth or Hayworth, and then the simplified Hayworth. I'm fine if you memorize these as either simplified or Hayworth. When you do that, though, be consistent. So in other words, if you draw the simplified Hayworth form, then you have to realize that the end of this straight line for us is a hydroxyl group. So make sure you put the lines in the right place and in the right direction. If you extend this line through the carbon, in other words, the corner of one of these cyclic structures, then that means you think there's also a hydroxyl group on the other end of that, which isn't the case. If you decide to use the Hayworth, make sure that you are also being consistent. So make sure that one end of your line has a hydroxyl group and the other end is denoted to be a hydrogen. So do one or the other. Remember, this is more like biochemistry rather than organic chemistry. In organic chemistry, the end of one of these lines would be a methyl group. But we're going to use kind of the biochemistry symbolism in this case. Okay? Now let's look at how this cyclic structure formed. The cyclic structure forms because of an interaction between the hydroxyl group on the highest number chiral carbon and the carbon in the functional group. So this would be the hydroxyl group that now interacts with this carbon of the functional group. Another way of saying the functional group here is the carbonyl group, or the carbonyl carbon. Remember that? C-A-R-B-O-N-Y-L, the carbonyl function. In other words, the aldehyde group is the glucose carbonyl function. The ketone group is the carbonyl group for fructose. So that's the carbon that is susceptible to this attack by this oxygen. It's susceptible because of the double bond. And so these molecules will tend to go 5 to 1 on the cyclic structures. Okay? So that's what they're trying to show here, the oxygen forming here. Now, when that happens, this hydroxyl group essentially shifts to this oxygen. So you lose that hydrogen. So now you have a hydroxyl group on carbon number 1. So now when you draw this in a cyclic structure, let's look at the simplified. We now have a hydroxyl group on carbon number 1 because the hydrogen that was part of this oxygen that did this is now associated with the oxygen that was an aldehyde on carbon number 1. Maybe you have to listen to that a couple times, and this is probably all review though. So don't forget that oxygen here. On the other hand, don't forget that this carbon number 5 here no longer has a hydroxyl group. That's why this line doesn't go all the way through. And this, obviously, this angled line here shows the CH2OH. So this is the carbon and the CH2OH here. So this also has an OH group. Okay? This asterisk shows the carbonyl function, the carbonyl carbon. So for glucose and galactose, the carbonyl group is always part of carbon number 1. Now, if you go from the Fischer projection to the cyclic structure, there's a couple easy rules to remember. One is, if the hydroxyl group is to the right in the Fischer, it's down in your Hayworth. So let's look at carbon number 2. The hydroxyl group is to the right, that's down in the Hayworth. In 3 is to the left, so it's up, and so on. Remember, there's not one on 5. Okay? So for the hydroxyl group, right equals down, and vice versa. For the hydroxyl group, left equals up. Now, the hydroxyl group on carbon number 1 in the cyclic structure for glucose can either be up or down. Just remember that. If the hydroxyl group is down in the cyclic structure on carbon number 1, down equals alpha. So this would be an example of alpha D-glucose. Now, sometimes when these cyclic structures form, that hydroxyl group is up. And it has to do with the position of this oxygen in the aldehyde before this cyclic structure forms. We won't go into all that. Oops. Just remember the hydroxyl group can be up or down. If I say draw beta D-glucose, then in the simplified Hayworth, you're going to flip that hydroxyl group to be up. That would be beta D-glucose. Okay? In the Fischer projection, you can either draw it like this, or you can just put your double bond oxygen here and your hydrogen here. So don't worry about representing the alpha or beta form in the Fischer projection, just in your Hayworths. Okay, I'm not going to go through galactose because it's basically the same. Structure is very similar to glucose. And again, so you can follow those rules. Now, if you look over here, there's a couple differences. One is because this is galactose, this hydroxyl group is up. That's the difference. Here's the hydroxyl group and the carbonyl carbon. And sorry, I should have mentioned this. Another term for carbonyl carbon can be the anomeric carbon. So in this case, this is beta D-galactose. So the beta indicates that the hydroxyl group in the anomeric carbon is up. That's all that means. So here's fructose, right? It's a ketone or a ketose. So its oxygen that's susceptible to nucleophilic attack is on carbon number 2. So now this ring structure forms between this hydroxyl group and that carbon. So here's our oxygen bridge. You can see that carbon number 1 is not part of this. So it ends up being kind of the tail to the ring structure, right? So now we draw it like this. Sorry, I lost my cursor. Now we draw it like this. Simplified version is here. Here's our oxygen bridge between carbons 5 and 2 in the case of fructose. Remember, here's our anomeric carbon. Here's the CH2OH as part of carbon number 1, so we draw it with a line and an angle. This line extends through here in the case of fructose because we have a hydroxyl group here. This is the hydroxyl group that determines whether or not it's alpha or beta, not this one. Okay? So in this case, the hydroxyl group is up. So this is beta D-fructose. Now this could be flipped, where you would draw where your line was going up and then your angle's here. If it's like that, then it's alpha, because in that case the hydroxyl group would be down. Okay? Now here we tend to draw it like this. Remember, we do not extend this line through carbon number 5. There's not a hydroxyl group there. And in this case we can draw that going up, and here's carbon number 6 and our H2OH here. It's typically the way we draw that. So that's beta D fructose. Simple enough. So an easy way to do is just kind of memorize and practice drawing the structure of the Fisher projection and the simplified Hayworth. That's the fastest, easiest way to do it. So again, that nomenclature, it's important because as we look through metabolism, we'll have to be able to identify those structures. Okay, so those are monosaccharides. Now how about if we combine these? If we combine these, let's talk about disaccharides and oligosaccharides. So disaccharides are obviously 2 monosaccharides linked together, and oligos are 3 to 10 monosaccharides linked together. And they're linked together through covalent bonds called glycosidic or acetyl bonds. I'll typically say glycosidic, that's a covalent bond. And those bonds can either have a configuration of alpha or beta, depending on its position before bonding, so we'll go through that. We'll also talk about a term called reducing sugars. These are the most common disaccharides. Here, for example, is maltose. It's glucose. And these structures aren't perfect. If I wanted to, we could kind of pick these apart, but let's kind of stick to the most important points of this. Here's maltose. So maltose is glucose. We already know that. Here's the glycosidic bond right here. Now, in the case of maltose, in order to name these glycosidic bonds, you first find the anomeric carbon or carbons. Here's glucose. So here's its anomeric carbon. It's in that glycosidic bond. Here's the anomeric carbon of this glucose. It is not in that glycosidic bond. So here's our bond. So we found our anomeric carbon. Now we use the number of that carbon. So in this case, we will say that's carbon number 1. And the hydroxyl group in this case was down when this bond was formed. Okay? Down equals alpha. So in the case of maltose, this is an alpha 1 to 4 glycosidic bond because this is carbon number 4. So maltose is alpha 1 to 4 glucose. Okay? You can see here that this says maltose in its beta form. And the reason is because this open anomeric carbon, the open anomeric carbon does not have a glycosidic bond. Its hydroxyl group can either be up or down. Doesn't matter, it would still be maltose. So if it's maltose but the hydroxyl group is up, we say that's beta. If this hydroxyl group was down, we would say alpha form. That's all that means. Okay? So that's maltose. Here is, and remember, it takes an enzyme to make those, but it also takes an enzyme to break them because they are covalent bonds. Here's lactose, one of the another very common disaccharide in our diet. We know lactose is galactose glucose or glucose galactose. Find the anomeric carbon. Here's the anomeric carbon for galactose. Here's the anomeric carbon for glucose. The anomeric carbon of galactose is in the glycosidic bond. So in this case, we would say, and that hydroxyl group was up when it formed the bond. So we know that this is beta form. So we know that that's beta. So in this case, the glycosidic bond for lactose is called beta 1 to 4 galactose glucose. Here's beta 1 to 4 galactose, glucose. Couple things. One is, I don't really care for the way they drew this because sometimes we see that angle, we say that's another carbon. It's not. So a better way to draw it is probably if you were to draw this more straight up and then have your oxygen group and then have this going into your carbon number 4, so it's more linear rather than having these angles. So there's not another carbon there, it's just the way they chose to draw it. And again, here's the anomeric carbon. It's open, it's not bound. So in this case, they said beta, but that's a typo, which are fun. In this case, the hydroxyl group is down. So this is the alpha form of lactose. Okay, now let's look at sucrose. It's unique. So here's sucrose, very common disaccharide, obviously. We know it's glucose, fructose. Let's find the anomeric carbons. For glucose, the anomeric carbon is here. For fructose, the anomeric carbon is here. So in the case of sucrose, both of the anomeric carbons are in the glycosidic bond. So there's a little bit of ambiguity in how we name that glycosidic bond. For sucrose, probably the most common way to describe it is alpha 1, comma, beta 2, glucose, fructose. Alpha 1, comma, beta 2, glucose, fructose. Right, alpha 1, it's alpha because it's down. And then here's carbon number 2, which is up. Alpha 1, beta 2, glucose, fructose is a common way to name the glycosidic bond of sucrose. Okay? Which ones are reducing sugars? In general, reducing sugars have open anomeric carbons. It's called a reducing sugar because in certain food chemistry reactions, this hydrogen can dissociate from the oxygen and donate its electron to another molecule and thereby reducing it. And so we say, oh, this is a reducing sugar. It's got an open anomeric carbon. This is a reducing sugar. It's got an open anomeric carbon. Sucrose is not a reducing sugar. Does not have an open anomeric carbon, does not reduce other molecules. That concept is probably more important in food chemistry than physiology. Okay, but that's where it comes from, so you know that. Okay, hopefully that's good. Review that if you need to. You can practice drawing these structures because you'll be asked to do that. Now let's go, next one, this is making a disaccharide. So in this case, we have glucose, glucose. And if you combine those, form that glycosidic bond, OH comes off here, hydrogen comes off here, that's water. So when you make a disaccharide in this case, you are generating water. That's called a condensation reaction. So making a disaccharide involves condensation reactions. If you look at these structures, this is glucose, look at the bond that's formed. It's alpha 1 to 4, glucose, glucose, that's maltose. Okay? And again, if we want to break that, it takes an enzyme. And hopefully, no surprise, that enzyme would be called maltase. We'll talk about that in a few minutes. This is how you hydrolyze these disaccharides. Remember, these are covalent bonds. So it generally in our body requires an enzyme. The enzymes need electrons. They can use the electrons from a water molecule in order to do this. So imagine this is maltase. Maltase, the enzyme actually grabs a water molecule and basically forces it into this glycosidic bond and hydrolyzes it. So this bond is broken. So now you end up with the hydroxyl group here and the hydroxyl group here. You've added water, and now we have glucose, glucose, which can be absorbed. So that's how that works. Let's talk about polysaccharides. These are polymers of repeating monosaccharides. Most common one we'll talk about is starch. Where starch is a homopolysaccharide. So the homopolysaccharide means that the polysaccharide is made from one type of molecule, which we know is glucose. So starch is all glucose. It's actually composed of two homopolymers. In other words, there's two different structures for starch. One is amylose, the other one is amylopectin. Here's amylose. And amylose is all glucose, glucose, all linked by alpha 1 to 4 bonds. That's it. So amylose tends to be this long kind of linear form of starch. Here's amylopectin. So these are both forms of starch. And in general, in plants, most of the starch is present as amylopectin. It tends to be about 80 to 85% of starch is composed of amylopectin. About the other 15 or 20% is amylose. Amylopectin has an additional glycosidic bond. So these are all glucose residues. Here's our alpha 1 to 4, alpha 1 to 4. This glucose is alpha 1 to 4, but it also has another glycosidic bond here. Here's our anomeric carbon. So we call this an alpha 1 to 6 because the oxygen bridge is the hydroxyl group that was bound to this carbon 6. So we say this is alpha 1 to 6. That's called a branch point. So now imagine this long structure of glucose molecules in alpha 1 to 4, and then you can also, some of them have alpha 1 to 6 branch points. And then you can have more alpha 1 to 4s. You would have this long sequence here, and then down here you might have another alpha 1 to 6. So it's kind of a combination of them. Okay? For starch, for amylopectin, the branch points end up being about every 25 monosaccharides. Here, if we look at it this way, maybe this is better. So here's polysaccharide comparisons. Here's starch unbranched, so we know that's amylose. You can see the structures here. Here's starch branched. So we'd have alpha 1 to 4s here, and then boom, here's an alpha 1 to 6. And for starch, it tends to be about every 25 glucose molecules you have a branch. All right? Here's glycogen. Remember, this is a glucose storage form in plants. Glycogen is glucose storage form in animals. So here we have glycogen is alpha 1 to 4, and a branch point, alpha 1 to 6. Glycogen has more alpha 1 to 6 bonds than amylopectin. The branch points in glycogen tend to be about every 5 to 10 glucose molecules, rather than the 25 of amylopectin. Okay? A couple of advantages to that. So glycogen tends to be more dense than starch. The other advantage of that is when glycogen is broken down, the enzyme starts on the end of the molecule and works its way inward. So the more ends you have, the faster that those enzymes can work to break down glycogen. And as you can imagine, it's advantageous to be able to go from glycogen to glucose if we are trying to perform high-intensity exercise. The more glucose, the faster, the better. So it's important for that. And the glycogen structure, because of all that glucose in a relatively small area, attracts quite a bit of water. So if a person depletes their glycogen in their body, they tend to also lose the water associated with it. So if you're minimizing carbohydrate intake and depleting glycogen, then the water, you also lose weight as water as part of that. Okay, here's cellulose. you can see right away these bonds look different structure. We know cellulose is a starch. And here's another way of looking at the structure of cellulose. You can see here, these are all glucose. So cellulose is all glucose. So it's a homopolymer as well. So let's find the anomeric carbon. Here's the anomeric carbon, right? So this is carbon number 1. Its hydroxyl group was up. So cellulose is beta 1 to 4, not alpha. Because it's beta, the enzyme recognizes the enzymes that we have that break down starch don't break down cellulose. So the beta bonds between glucose residues cannot be hydrolyzed by the digestive enzymes of humans. So for us, cellulose is a fiber. Now, kind of unfortunately for us, cellulose is considered probably the most common organic molecule on Earth. It's a huge source of glucose, but not for us. Some organisms can get glucose from it, but we don't. Bacteria can break that down, and so animals like cattle, for example, have bacteria in their gut, so they can eat grass or hay or something like that that has cellulose in it, and the bacteria break down those bonds, and then they obtain glucose, and then the bacteria make short-chain fatty acids, that they basically make some molecules that provide energy for the cattle or the ruminants. And so that's how they get it. If we could get glucose from cellulose, we could basically eat grass and get glucose. Fortunately or unfortunately, that's not the case. So I think that's all I want to say there. So let's go to the next one. Let's go to Carb Lecture 2. It should be here. And so let's kind of switch gears here. So in Carb Lecture 2, sorry, I'm drinking a little bit of caffeine. This is the outline. Phases of carbohydrate digestion, we'll talk about that. The enzymes and their substrates, absorption of monosaccharides, and then we'll go into the circulation and regulation of blood glucose. So we're going to kind of follow this pathway on how we obtain glucose and then how it's metabolized. Okay. There are 2 phases of carbohydrate digestion. So we're going to talk about that first. And I have a table, I think, on the next slide. And then the second bullet point to this, we'll come to this in a second. That's what is the key enzyme for starch digestion. So we're going to kind of put all this together. So I drew this on the whiteboard in here. That's why it doesn't look very good. That's my writing. And a few points from this. So if we look at the left side of this table over here, we can see this is the food source. So these are the major carbohydrate sources in our food. We have starches, which includes both amylose and amylopectin. We're optimistic, and we're going to say that people consume approximately 60 to 70% of their carbohydrates in the form of starch. Okay? Here's a second very common form of carbohydrate, and that's lactose. You know, lactose is in dairy products, in milk, and maybe a little bit in cheese, and some in a little bit in in yogurt. And then down here at the bottom, we're going to say that's 0 to 10%. And then here's sucrose. So sucrose, we're going to say, is about 30%. And I'd say in general, you know, people should consume probably 25% or less of their carbohydrate. But I mentioned this before, a lot of people consume 50% of their carbohydrate as sucrose. So we eat way too much sugar in this country in general. So sometimes one of the best things a person can do to their diet is decrease sucrose. Okay? All right, so we have that over here. Now let's look at this. Here are the 2 phases. Phase 1 is luminal hydrolysis. In other words, some carbohydrate is hydrolyzed in the lumen of the small intestine. So carbohydrate goes in, it's in the lumen of the small intestine, and an enzyme degrades it. In this case, that takes starches from these big structures of glucose, to maltose, which we know is a disaccharide, maltotriose, which is a trisaccharide, and alpha dextrins. We'll talk about their structure in a second. Okay? You can see that in the luminal phase, lactose or sucrose do not require or do not undergo any enzymatic hydrolysis. So these are two disaccharides. Nothing happens to them in the lumen. And yet they can't be absorbed in that form. Remember, carbohydrates are absorbed as monosaccharides. So we need another step. So now these molecules contact, oops, I forgot to underline phase 2. This is phase 2. This is what happens on the brush border membrane. So this is brush border membrane hydrolysis, phase 2. These glucose-containing molecules are converted to glucose. They're all hydrolyzed to glucose. These disaccharides are converted to glucose and galactose. Sucrose is converted to glucose and fructose. So now we're all monosaccharides, and they can be absorbed. Okay, so it takes 2 phases and a few different enzymes. So let's go through that. So this is starch digestion. And the second part of this is, what is the key enzyme for starch digestion? That is pancreatic alpha-amylase. And you can see in this diagram, for example, in the lumen, starch is converted to these molecules. So pancreatic alpha-amylase works in the lumen of the small intestine, and it composes phase 1 of carbohydrate digestion. Because you can see here, here's the products of pancreatic alpha-amylase activity on starch, maltose, maltotriose, and what are called alpha-limit dextrins. And these alpha-limit dextrins are also known as alpha dextrins. I have that on the table. Also referred to as isomaltose. And so each of these terms refers to molecules that are 3 to 5 glucose molecules linked through alpha 1 to 4 and alpha 1 to 6 bonds. In other words, alpha-limit dextrins have both alpha 1 to 4 and alpha 1 to 6 bonds, but they only have about 3 to 5 glucose molecules. So imagine amylopectin being reduced all the way down, going from hundreds or potentially thousands of glucoses linked together to 3 to 5. Okay? Can't be absorbed yet, but these molecules are much smaller products than starch. So here's pancreatic alpha-amylase. This is an interesting enzyme. This is its active site. So this kind of filled in rectangle shape here, it says A through E, represents pancreatic alpha-amylase. It has 5 binding sites. This enzyme only works efficiently if all 5 of its binding sites are filled. Okay? So this enzyme only works efficiently when all 5 of its binding sites are filled. Once they are filled, the enzyme will cleave at one location, and that's what's shown here. What's filling those binding sites? These are glucose molecules. And you can see here, this would be, it's pretty linear, so these would be alpha 1 to 4s. Pancreatic alpha-amylase only cleaves alpha 1 to 4 bonds. So it would cleave here. And then once it cleaves, it lets go. So both sides or parts of this molecule would then be released. And then this enzyme would now look for another location on starch where it can fill all 5 of its binding sites and cleave again. So that's what it's going to do. Okay? So, sorry. you look at this end of the molecule, this means that the carbon number 4 is open. In other words, this is one end of it. And this shows repeating glucose molecule, unknown number here. This diagonal line through this means that this is the reducing end. So in this case, carbon number 1 has an open anomeric carbon. That's all that means. Okay? So pretty interesting. So when we consume starch, that's the enzyme that we rely on. I'll try to remember to mention this again later, but this is one of the main reasons why we wait in infants for their pancreas to become fully functional. And that typically happens at about 6 months of age. Before 6 months of age, we don't give infants starch or other foods like it because the pancreas is not up to kind of a maximal capacity. Once we get to that point in life, our pancreas can make enough pancreatic alpha-amylase so that the starch can be hydrolyzed and so that we can actually go through this process. The enzymes on the brush border membrane are at maximum level before birth. So when an infant is born, they can break down lactose. And you can see why that would be important, right? That's their main carbohydrate source is lactose from milk. So they have to be able to have lactase. And then interestingly enough, as we get older, it's very common for lactase to become reduced in its levels. And when lactase becomes at a lower level, that's when we can develop what's called lactose intolerance. And that means that the lactose is no longer degraded at a high level, and the lactose goes through the small intestine and goes into the large intestine, and the bacteria in the large intestine can metabolize the lactose, no problem. They love it. The bacteria make molecules that attract water into the gut. And so if a lot of lactose goes into the large intestine, it can cause abdominal pain, it can cause bloating, it causes gas production, all of that. It can cause what's called osmotic diarrhea, and that's why. And especially in certain ethnic populations, that's very common. Certain populations are not uncommon for 80 or 90% of the people in that population to be lactose intolerant. And it typically a lot of times will happen when a person's about 18 or 20 years old, something like that. They'll realize, oh, it's hard for me to drink milk. Sometimes it's mistaken for a food allergy or a milk allergy or something. Sometimes it's as simple as lactase expression is now at a lower level. And we won't go into the mechanism, but it's actually kind of a default where the system changes the way the transcript or the mRNA is spliced, and so you end up with less lactase on the surface of the brush border membrane, and so you lose the ability to degrade as much lactose that was previous. It typically stays that way. In other words, once a person's lactose intolerant, it tends to be that way. So there's options. You can buy lactase enzyme and consume it before a meal or with a meal, and they've made those enzymes so that they can actually go into the small intestine and be functional, and then they can break down the lactose so that it can be absorbed as glucose and galactose. There's also products like Lactaid, the milk. In that case, they've treated the milk with lactase before it's sold, and so if you drink Lactaid milk, it doesn't have any lactose left in it, but it does have glucose and galactose, so Lactaid milk is actually a little bit sweeter than regular milk. And if you think about it, unfortunately those products are expensive. The Lactaid, and you have to take lactase pills. They cost money. Lactaid milk is probably about twice what normal milk costs. But the trade-off is if a person shies away from dairy products because they're lactose intolerant, they lose a huge source of calcium. It's very difficult to get adequate amounts of calcium, especially if a person's avoiding dairy. So that's a consideration. Unfortunately, kind of indicates another potential socioeconomic problem. And also milk is a good source of potassium and a good high quality protein. So unfortunately, that can be a problem, and it hopefully can be considered. But that's a little bit about the physiology and the temporal expression of that enzyme. Okay, so let's go back to pancreatic alpha amylase. This enzyme we know is the key one for starch, and this diagram isn't perfect, so let's not get too hung up on it, but this, for example, is the structure of amylose, and we can see this means it continues. Here's the reducing end, and here's amylopectin, and this is showing that this is a cleavage site. So in other words, if amylase finds this site on amylose, it can bind here, imagine it binding here, filling all 5 binding sites, and then cleaving here and then releasing. It'll release, in this case, maltotriose and maltose, which could then be cleaved on the brush border membrane. If we look in this case over here, let's say this is amylopectin. The problem with amylopectin is these alpha 1 to 6 bonds prevent amylase from filling all 5 of its binding sites. So this is not linear. So this is steric hindrance. You've probably heard that term. Amylase can't cleave in this area. So amylose can bind over here, it could bind over here, it can cleave pretty close to the alpha 1 to 6. It could cleave here, it could cleave here, but you can see what it leaves. It leaves this 5-glucose molecule that has both alpha 1 to 4 and alpha 1 to 6. And that's what's called an alpha-limidextrin or an alpha-dextrin. So you end up with these molecules. And this molecule needs to be degraded by a different enzyme. Okay? So it's very common. We said the most common form or structure of starch is amylopectin, and the most important enzyme for starch digestion is amylase. So you end up with a lot of these alpha-limidextrin molecules in the gut. For example, up to 1/3 of the mass of the final breakdown products of amylopectin are alpha-limidextrins that need an additional enzyme in order to be for their next step. Okay, so here is the kind of phase 2. So we talked about amylase, and now we're on phase 2. So in this case, we have our hydrolases on the brush border membrane. They finish starch digestion. They also cleave lactose and sucrose, the other major carbohydrate sources. And they are sometimes referred to as oligosaccharidases, right? Enzymes that cleave oligosaccharides. And I think I have a couple more slides to describe these. There's several types of these enzymes. One category or type are called alpha glucosidases. And as their name implies, they target alpha glycosidic bonds and glucose molecules contained within those molecules. So the most common alpha glucosidases are sucrase, isomaltase, and maltase. And sometimes their name is direct reference to their substrate. Sucrase cleaves sucrose, for example. And there's also another enzyme called a beta-galactosidase. Right? So rather than an alpha-glucosidase, this is a beta-galactosidase. And as its name implies, it targets molecules that have a beta-glycosidic bond and contain a galactose. And that happens to be lactase, that enzyme. So let's look at this. I have a table here. Not a perfect table, but hopefully it helps. This is a few of these enzymes. We're going to focus on 4 of them. The first 3 here are alpha-glucosidases. The last one is a beta-galactosidase, and that's lactase. We talked about lactase, and its substrate is lactose. Here's sucrase. Now, sucrase is interesting because its principal substrate is sucrose, but sucrase can cleave sucrose, but sucrase can also cleave maltose and malto-oligosaccharides. So sucrase is an enzyme that can hydrolyze more than one type of glycosidic bond. Remember the sucrose we said is, its chemical bond is alpha 1 beta 2 glucose fructose. So it cleaves that one, but it also cleaves alpha 1 to 4 links of maltose. So it can also cleave maltose. Okay, so interesting. The reason they call it sucrase is because it's the only one that cleaves sucrose. Second one is isomaltase. And this one you want to put a star by, because this is the only one of these enzymes that can do alpha 1 to 4 and alpha 1 to 6. And remember, these are what we call the alpha dextrins. They would have alpha 1 to 4 and 1 to 6. Also referred to as isomaltose. Alpha dextrin and isomaltose are the same. So we call this enzyme isomaltase. So it's crucial, but it breaks down those alpha dextrins. And then you have maltase. As its name implies, it'll degrade maltose. And if it, for example, it's a maltotriose, maltase can also work. So maltase cleaves alpha 1 to 4 links only. In other words, doesn't cleave alpha 1 to 6. Okay, and remember, these enzymes are all in the brush border membrane. Lactase, the last one, is also on the brush border membrane, because that's what we're talking about here, phase 2. These enzymes are on the brush border membrane. So let's do a couple more. This is an interesting slide for a few different reasons. One is it kind of shows how two of these brush border enzymes get there. And so this is looking at the synthesis of sucrase isomaltase, two different enzymes that are included in this table. These two enzymes are actually made from one gene. So they're made from one mRNA. That mRNA is then translated and goes through the secretory pathway. So the mRNA starts translation, has a signal peptide, and then the peptide is targeted through the secretory pathway. In the very last step, that protein is cleaved or cut that yields two separate functional enzymes. So one gene, two enzymes. We're looking at this process in some of the later steps of the secretory pathway. So you should be able to go from the gene to the final proteins. Okay, so don't put this slide on its own. You want to be able to put it into the bigger picture. In this case, we have an experiment that was done a long time ago in rats where they showed the time course of how long it takes for it to go through the ER to the brush border membrane. That's all that means. Okay, so imagine, and this is an intestinal cell. We're not looking at it in its entirety. So we're missing the nucleus, we're missing the first part of the RER and all that. This kind of light gray line is showing the protein or peptide. The dark lines here are showing carbohydrate being added. So in this case, this protein is in the ER. More correctly termed, it should be the RER, the rough endoplasmic reticulum. It's been made, it's inside there, and now it's starting its glycosylation. Goes from the RER lumen to the Golgi, where more carbohydrate is added. Remember, we said that's typically what happens in the Golgi, folding, glycosylation. So now more is added. That's why they're showing it like this. And now that Golgi is moving towards the plasma membrane. So now we have a transport vesicle. And this protein is embedded in the brush border membrane. About 95% of the protein extends into the lumen of the small intestine. Here's the lumen of the small intestine. 5% of the protein embeds in the membrane to hold it there. So this is a brush border membrane enzyme that stays there. The enzyme's not functional yet, so P stands for precursor. It's in place but not functioning. So it's been inserted, but now it needs to be processed. So this is known as post-insertional processing. The processing is when trypsin from the pancreas cleaves the precursor into two functional enzymes, isomaltase and sucrase. And this is a bad diagram. Sucrase is still bound to that brush border. But now you have isomaltase, which can cleave isomaltose, and sucrase, which can cleave sucrose. Okay? So again, precursor's embedded. Trypsin is a protease from the pancreas that has quite a few targets, including this protein. So trypsin is a protease that comes from the pancreas. Trypsin digests dietary protein, but trypsin also cuts this precursor so that it becomes two functional enzymes, isomaltase and sucrase. And that's how they end up in that brush border membrane. Pretty interesting. One gene, two functional proteins that are made with a post-insertional processing mechanism. And that's how they get there. So here's a summary of what's going on, kind of a big picture. Major carbohydrates here, we talked about this. Here's starches, which require this first additional phase 1 step. So this is pancreatic alphaamylase. Generates these products, which are then further hydrolyzed in step 2, phase 2, to generate glucose. Here are the major disaccharides in the diet. They have a single step hydrolysis involving these enzymes that are on the brush border membrane to generate these monosaccharides that can then be absorbed. Okay? So I'm going to stop there because we kind of changed topics a little bit.